Why should we study microbiology? Well, microorganisms are found everywhere, in our food, out in the ponds, in the soil. They cause us problems. Most of the time, they're very beneficial. About 95 to 99% of all microorganisms are good guys. They're beneficial organisms, and they're supposed to be here on Earth. If you take a look at the image of the pool on the left-hand side of the slide, it's green in color because of the growth of microorganisms. The fire in the photo in the middle of the slide is due to the decomposition of organic matter out in the forest that released a combustible gas called methane, and with an ignition source, caught fire. So now someone has to put that out. And then the two photos on the right show pools or sources of water that are pink in color. These are actually saltwater pools or saltwater sources with a pink-colored microorganism that's growing in this very salty water, used, especially in the photo on the bottom, to manufacture salt. Now, if you launch PowerPoint, if you haven't already, and click on the little link that says microorganisms, so the word microorganisms is actually a link to an alternate website, there's an image that you can enlarge to see just how big are microorganisms. It's actually a web page called Cells Alive, and they have some very interesting, fun things to do with images and different kinds of microorganisms. Here's a specific example of a type of microorganism that's pink in color and grows in salt water, which is the reason that the photos on the previous slide of the salt pools, the bottom photo on the previous slide is salt manufacturing, where Halobacterium is growing in that salty water, and the top photo that's pink is just simply a naturally occurring brine pool with Halobacterium growing. Halo is an ancient term that means salt or briny, and so Halobacterium would be considered a halophile, in other words, a salt-loving organism. The second name for this organism, Salinarum, includes in the word the ancient Latin term that essentially means salt. We also use that sal or sally as part of our word salary even today, because years ago in ancient Rome, people were paid in salt. That was their salary. Now, back to Halobacterium. As a halophile and a salt lover, this organism has specific growth requirements. It's actually categorized or grouped in a group of microorganisms we call archaea. We'll describe that in a little more in just a few slides. But with Halobacterium, that pink color that we see inside of the cells is actually a biochemical called bacteriorhodopsin. Bacteriorhodopsin can sense and detect light and allows Halobacterium to adjust its location in the water so that it's receiving just the right amount of sunlight. Bacteriorhodopsin is visual purple for the microorganism. If you've ever heard of rhodopsin that's in the human eye, which is called visual purple in the human eye, it's sensitive to photons of sunlight. When you shine light on either rhodopsin or bacteriorhodopsin, you'll see what happens on the right-hand side of the slide is that the bacteriorhodopsin and rhodopsin, whether it's in the archaea, bacterial Salinarum, or if it's in your eye, it actually changes color. It goes from this sort of purplish pink to a clear color, and then after a little bit of time, it goes back again. So a light-sensing pigment or light-sensing biochemical. If we're working with bacteriorhodopsin from Halobacterium, it's a good research tool. It gives us an idea of some of the fundamental biochemistry, and then we can go into, let's say, the human eye and look at the biochemistry of rhodopsin from the human eye and learn a little bit more. And another kind of biochemical that's found in Halobacterium that's an important research tool, and again model, both of these biochemicals are models that we study, then go to human sources and compare the differences and the similarities between those biochemicals. And Halobacterium also has a heme protein inside of its cells that sense oxygen. very similar to the heme protein in our red blood cells that we call hemoglobin. Hemoglobin in our red blood cells is responsible for capturing oxygen in our lungs and then transporting that oxygen to the cells in our tissues. So again, a good way to study some of the basic biochemistry and the basic science of chemicals that can detect light and chemicals that sense oxygen is to use the archaea halobacterium. Deinococcus radiodurans is another example of microbial diversity like the Halobacterium salinarum. Both Halobacterium and Deinococcus are extremophiles. They will grow in extremely challenging or difficult environments here on Earth. For Halobacterium, it's a very salty environment. For Deinococcus, any place there might be extremely high energy ionizing radiation. Both of these extremophiles, in other words, challenging, extremely difficult growth conditions, have their own unique characteristics, and Deinococcus is one that can tolerate extremely high levels of ionizing radiation. 500 to 1,000 rads of ionizing radiation could be enough to kill a person depending upon the location that your body is exposed. Think of ionizing radiation just like something that's going to blow up a cell. It's very, very dangerous depending on how much and where it's exposed on your body. Well, Deinococcus, you can expose Deinococcus to up to 1.5 million rads of radiation and it will still thrive. Doesn't get destroyed by just extremely high doses of ionizing radiation, which most other organisms would. Magnetospirillum magnetotacticum is another example of microbial diversity. It's a gram-negative bacterium, which will make more sense as we work through the information in Unit 1. It has some means of movement that we call motility. It's microaerophilic. Micro means small or tiny. Aerophilic, well, if you think about it, it's referring to air. So this organism grows just under the surface of the water or just under the surface of the soil, where there's just a little bit of oxygen, not the typical kind of oxygen levels we expect on the surface of the Earth. It also has a spirillum shape, in other words, a gentle S-shaped curve to the individual unit structures. The picture that's on the slide is a photo of a single magnetospirillum cell or organism. If you look inside, you can see these little dots, little dark blackish looking dots. Those are concentrated sources of iron that have magnetic qualities. The little dots, each one is called a magnetosome, and collectively in that little sort of chain or string that we can see inside the magnetospirillum cell are magnetosomes. Now, that line of magnetosomes can act very much like a compass needle and allow the organism to align itself with the different magnetic fields in the Earth. We don't have very many common names or everyday names for microorganisms. We'll spend much of this semester learning scientific names of unique or specific examples of microorganisms. But the few common names we might have, bugs, germs, well, a better scientific word or actually medical term meaning the same group or type of organism that causes us harm would be a pathogen. A pathogen is a microorganism that causes disease. Well, those are the ones that catch our attention. The photo in the lower right of the slide is an image of the hand of a patient with septicemic plague. Septicemia means we have bacteria or a microorganism actively growing in the bloodstream. Now, unfortunately, the organism that causes this infection, which was septicemic plague, is Yersinia pestis. Yersinia pestis triggers the production of small, tiny blood clots. Those tiny blood clots get into the tiny blood vessels in our body, like the fingertips of the hand, the tips of the feet, nose, ears, just any place that there are small, tiny blood vessels that we call capillaries that can get occluded or plugged up with these tiny blood clots. Well, eventually we don't have good blood perfusion of those tissues, and the cells in those tissues die. So that's why the fingertips of this patient are black. The only thing that we could do for this patient is to essentially amputate all of that dead flesh so they wouldn't become susceptible to additional kinds of problems. Those are the types of microorganisms that catch our attention, the pathogens like Yersinia pestis. The image on the right, I've tried to include an image of a normal healthy brain in the lower right so you can get an idea of the contrast between what a normal healthy brain should look like on autopsy versus what happens if someone has hemorrhagic meningitis due to inhalation anthrax. Bacillus anthracis is the pathogen that causes hemorrhagic meningitis. That discoloration or dark color over the surface of the brain is not normal. It's actually due to blood vessels that have ruptured and leaked blood all over the surface. And actually, if you can kind of look inside where the cutaway is on this slice of brain, you can even see the blackening on the inside. So pretty much anywhere and everywhere that the blood vessels could rupture in this patient's brain, they did. What are microbes or microorganisms? Our first category of organisms that we call microorganisms are the viruses. Now, viruses are essentially acellular in structure. If you describe an organism as having cells as their fundamental unit or fundamental structure, then a virus truly doesn't fit that category or that description because they are acellular. Viruses have to get inside of a cell in order to grow and reproduce. Now, as microbiologists, we have no problem describing a virus as a microorganism because we're actually going to use slightly different criteria than, let's say, a classically trained biologist would use to define an entity as an organism. As we work through Unit 1, we'll have our descriptions of the criteria that we use as microbiologists to describe microorganisms. We seldom, if ever, use the word living or alive. And you'll find why as, again, we work through this unit. The second group of microorganisms are the bacteria. Bacteria do have cell structure. They have a cell structure that we call prokaryotic cell structure. Now, if you don't know what that means right now, we will go into detail about different kinds of cells that we find here on Earth. But needless to say, prokaryotic cell bacteria are commonly found in all kinds of places on Earth, and we will spend a lot of time in lab working pretty much just with bacteria. If you take a look in the image on the lower left, there we have colonies of bacteria growing on something that we call agar. Agar is a gel to which we can add different kinds of nutrients. When we pour that gel into a device called a petri dish and allow it to set up just like a piece of Jell-O, even though it's not exactly the same as Jell-O, and then we can spread or place different materials, specimens onto the surface of the agar and allow any bacteria that might be present in that specimen to grow, the next thing you know, after 18 to 24 hours typically, we find what look like little drops of paint or little colored drops of something growing on the agar. Those things are colonies. Most of the images that you're seeing, or the little dots that you're seeing on the agar plate in this photo, are colonies of bacteria. The larger blackish or charcoal colored colony at the bottom of the plate, or the petri dish, is actually a colony of mold. Now, in lab, we're going to take little samples from these colonies, which we call macroscopic or colony morphology, or images, and then we're going to spread a little bit of a sample from each one of these dots or colonies onto a glass microscope slide, stain them in a procedure that we call a gram stain technique to color the little cells from the colonies to determine the microscopic morphology. Microscopic morphology, what does it look like through the microscope? Macroscopic colony morphology, what does it look like on the surface of an agar plate? The archaea would be the third group of organisms that we call microorganisms. They are also prokaryotic in cell structure. Ages ago, a few decades ago, scientists thought these were simply bacteria that grew in very extreme environments, so we called them archaebacteria. After thoroughly studying their structure and biochemistry, it's now been determined that this is actually a distinctly different group of microorganisms. The archaea, even though they have prokaryotic cell structure, are unique, almost as an example of what's called parallel evolution. We take a look in very salty, briny pools of water, very hot boiling water pools out in, let's say, Yellowstone National Park, as we see in the lower right-hand photo, or if we go down to the bottom of the ocean, where we have hydrothermal vents that are seeping huge quantities of geothermal energy in the form of heated water and dissolved minerals, which are the foundation for a food web based on archaea that are living and growing on the inorganic dissolved minerals in this very hot water. Scientists were astounded to find that archaea living from the minerals or on the minerals in this very hot, high-pressure environment was the foundation of a food chain or food web surrounding these hydrothermal vents. Using the archaea as that primary source of food, much larger animals like clams, shrimp, crabs, and even the very large tube worms, which are those long sort of stick-like or rod-like structures in the photo on the top right, the red pigmented color that you see sticking out from those tubes are actually the archaea growing in the flesh of these very large worms. A typical tube worm closer to the surface of the earth is probably about 4 centimeters long. These tube worms at the bottom of the ocean are about 1 meter long, so about 10 times or maybe even larger, hundreds of times larger than your typical tube worm, all based on using archaea as a source of food. A third type of prokaryotic celled microorganism would be the cyanobacteria. The cyanobacteria are the greenish or blue-greenish, slimy little microorganisms that are growing in the ponds in the spring and the summer. If you see something really dark blue-green, shiny, again, sort of slippery if you tried to collect or grab a sample, those are the cyanobacteria, capable of capturing sunlight and converting the use of that sunlight energy with some carbon dioxide, those kinds of materials into more usable materials for the rest of us. Cyanobacteria used to be called blue-green algae, and so typically in textbooks you'll still see both names used for this group of microorganisms. Another group of microorganisms are the protists. The protists involve a huge, diverse group of organisms, ranging from diatoms to amoeba to paramecium to rotifers, all kinds of single-celled or multicellular, tiny, tiny little things that grow in the ocean, grow in ponds, and we generally collectively call them protists. If you've ever heard of a protozoan, like the amoeba, that's an example of a protist. Now we're seeing in the protists a uniquely different kind of cell structure that we call a eukaryotic cell. The eukaryotic cell is very similar, exactly kind of the cell that we find in the human body, so there are some distinctive differences and similarities, but fundamentally they're all going to be eukaryotic cells. The protists, so diverse in their structure and function, would be another group or category of organisms that we could call microorganisms. Another group of microorganisms would include the algae. They also have eukaryotic cell structure. They're all going to have some kind of color, ranging from golden to green to reds and browns, but collectively, algae are also considered to be microorganisms. Another group of organisms that are eukaryotic in cell structure that also include examples of microorganisms are the fungi. Now, if you've ever seen apples that have fallen on the ground in the fall and then they start to rot, well, apple rot is due to a microorganism that's essentially breaking down or decomposing those apples that didn't get eaten. If you've ever seen the fuzzy stuff that's growing on the forgotten food in your refrigerator, those are molds that are essentially breaking down the bread or food that you forgot to eat. And then if you've ever seen mushrooms, mushrooms are also classified as fungi, even though they're really not microorganisms. But we find examples of microorganisms in the group that we call fungi. Last but not least are the helminths, better known as worms. Some of the worms are microscopic. You need a microscope to see them. Some of the worms are macroscopic, like the photo on the right with the piece of intestine that's loaded full of adult worms. We will use a microscope to frequently identify the infective stages of these worms, whether the adults are large or not. And so within the scientific study of microbiology, the study of parasitic worms or helminths is also included.